Hello, welcome to this video about building a culture of responsibility among life scientists. My name is Michael Imperiali. Let me begin by telling you a little bit about myself. I am a professor of microbiology and immunology at the University of Michigan in the United States. My laboratory studies a virus called BK polyomavirus that is a significant problem in transplant recipients. I have been interested in the safe and responsible conduct of research for many years. Of most relevance to this video are my experiences during the last decade at both the international and national levels. Here at the University of Michigan, I am involved in our course for students and postdocs about the responsible conduct of research. I have been a member of the National Science Advisory Board for Biosecurity, or NSABB, in the United States since its inception. I have also served on National Research Council committees here that have addressed various aspects of the intersection of science, security, and public policy. Finally, I have been involved in international discussions both through my service on NSABB as well as through events organized by the National Research Council and the American Association for the Advancement of Science. My hope is that I can share with you some of the collective wisdom that I have garnered from my interactions over the years with many talented scientists, policymakers, students, and others. The opinions that I state in this video are my own, however, and do not necessarily represent those of these organizations or other persons. I will begin the video with a brief discussion of the importance and scope of the life science research enterprise. Next, I will take you through a brief history of the misuse of pathogenic organisms. Then, I will introduce the concept of dual-use research in the life sciences and talk about the discussions surrounding this issue both in the US and globally. I will use the recent controversy regarding the publication of experiments aimed at understanding transmissibility of H5N1 influenza virus as a case study of the dual-use issue. Finally, I will close with my thoughts about our collective responsibilities as scientists and educators and how we might move forward. The importance of research in the life sciences to our lives in the 21st century cannot be overstated. Just 100 years ago, the average life expectancy, even in the developed world, was very low due to disease. Particularly in the area of infectious diseases, we have made great progress. Although there is still a long way to go for some diseases and in some areas of the world, our ability to quickly identify infectious agents and develop vaccines and therapeutics is unprecedented. An example of this is the emergence of SARS in 2002. The virus was identified rapidly, and public health measures prevented what could have been a serious pandemic. Similar advances have improved the health of the animals and crops that we raise for food and of domestic animals. Moreover, the benefits of the tremendous amounts of knowledge that we accumulate, while they sometimes do not become obvious until years in the future, cannot be overstated. Therefore, the worldwide public is dependent on the work that we as scientists do to improve their lives every day. To me, one of the beautiful aspects of life science research is that almost anyone can do it. For the most part, it does not require expensive equipment or supplies. Therefore, one can start a biology laboratory much more easily than in other fields of science and engineering. In addition to the items that are needed to experiment, protocols and troubleshooting capabilities are largely available on the internet. I just did a Google search for how to set up PCR and got over 9 million hits. In addition, many scientific journals are making articles available for free. While this sometimes entails a bit of a delay from the date of publication, there is still a lot of information out there. Finally, governments recognize the importance of the life science research enterprise because of the reasons that I outlined earlier. Even in times of austerity, there is a significant public investment in research. Once again, this is a reflection of the role of governments in promoting the well-being of their citizens. So let's go through a little bit of history. Because they cause disease, there is a long history of the use of biological agents as weapons and to terrorize populations. During the Middle Ages, it is thought that the Tartars hurled bodies of individuals who died from the bubonic plague over the walls of their enemies, the Genoese, to spread the disease. Presumably the fleas that act as vectors to transmit the bacteria were along for the ride. During the colonization of North America, the British gave smallpox-infested blankets to the Native Americans. Since this native population was completely naive to the virus, the loss of life was very large. In modern times, there have been state-sponsored bioweapons programs. Both the United States and the Soviet Union had large efforts in this area during the Cold War. In 1979, there was an accident at a Soviet bioweapons facility in Sverdlovsk. Anthrax spores were released into the atmosphere. Scientists were able to reconstruct the spread of the spores as shown on this map based on weather reports from that day. Non-state actors have also attempted bioterrorism. In Japan in 1993, a cult known as the Aum Shinrikyo tried to release anthrax spores deliberately into the Tokyo sky. Fortunately for the citizens of that city, the group did not know its Bacillus anthracis genetics and used an attenuated vaccine strain. These photos are said to show aerosols being released from the cult's headquarters on the day of the attempted attack. The United States has not been immune to bioterrorism. In 1984, followers of Bhagwan Shree Rajneesh in Oregon tried to influence the results of a local election by contaminating salad bars at area restaurants with Salmonella bacteria, with the idea of making people too sick to vote. While over 700 people fell ill, the plot failed. Of course, the most famous attack on U.S. soil was in 2001, when anthrax spores were mailed to some U.S. senators and other individuals. The individual who was accused by the FBI, Dr. Bruce Ivins, committed suicide before he could be brought to trial. Coming on the heels of the September 11th terrorist attacks, the anthrax mailings heightened the awareness of the U.S. government to the threat of bioterrorism. The National Research Council, which is the operational arm of the U.S. National Academies, convened a committee in the spring of 2002 to look at the possible misuse of life sciences research in the 21st century. The so-called Fink Report, named after Jerry Fink, who chaired the committee, laid out the scope of the problem and made a series of recommendations to the U.S. government on how to proceed. There are two things in this report that are especially pertinent to our discussion. First, it gave a new definition to the term dual use. Previously, this term had been used to refer to technologies that had both military and civilian uses. The NRC report, however, used it to mean any results or technologies that could be used for either the benefit of mankind or be misused to cause harm. Second, it recommended that the U.S. government create an advisory panel to provide guidance to the government on the dual-use issue. This panel, the NSABB, was formed in 2004 and had its first meeting in June of 2005. I do not have the time to go into all the details of the NSABB, but I do want to focus on a few key points. First, the board has representation of experts from a diverse set of fields, ranging from practicing scientists to members of the public. Second, it has addressed various issues, including synthetic biology, communication of the results of dual-use research, and personnel reliability in research laboratories. the latter two of which are most relevant to this video. Third, since the beginning, NSABB has recognized that any efforts to deal with the dual-use issue cannot be confined to the United States and must be international in nature. As such, the NSABB has reached out globally and sponsored a number of activities with the international scientific community. You can find recordings and other information related to these activities, as well as other items of interest from the NSABB at its website, whose URL is shown here. One thing that the NSABB did was to recognize that the new definition of dual use proposed by the NRC committee was too broad. Almost anything can be misused. It therefore sought to come up with a stricter threshold with which to define research that is likely to pose the biggest threat. The NSABB definition states that dual-use research of concern, that is, the dual-use research about which we should be most worried, is research that, based on current understanding, can be reasonably anticipated to provide knowledge, products, or technologies that could be directly misapplied by others to pose a threat to public health and safety, agricultural crops and other plants, animals, the environment, or material. The key aspects of this definition are the immediacy and ease with which misuse can occur. How well does this definition work? It was put to the test at the end of 2011 when the U.S. government asked the NSABB to provide recommendations about the publication of 2 manuscripts involving the H5N1 influenza virus. Let me first give some brief background about the viral biology and then talk about what happened. Every year, there are generally subtle differences in the circulating strains of influenza viruses around the world. These differences are due to antigenic drift, or small numbers of mutations that lead to slight changes in the antigenicity of the virus. These new strains usually do not cause severe disease or result in pandemics because the population has some cross-reactive immunity. Sometimes, however, a completely new antigenic variant can make its way into the population. This usually occurs due to reassortment, or shuffling of the RNA genomes of 2 different viruses, often in an animal host. The H1N1 virus of 2009 is an example of antigenic shift. If the human population is immunologically naive to these new antigens, as it was to a large degree in 2009, such a virus poses pandemic potential. In 2003, reports of human infections with the H5N1 strain of influenza began to increase. This strain is known as a member of a class of influenza viruses referred to as highly pathogenic avian influenza because of their virulence in birds. The human infections with this strain were very severe, with a case mortality rate of approximately 60%. Fortunately, it appeared that this virus could not be spread from human to human. Almost all of the cases identified could be traced to direct contact of the infected individuals with birds. Public health authorities feared, however, that if this virus were to gain the ability to spread directly among humans, either through antigenic shift or antigenic drift, it could cause a pandemic of unprecedented lethality. As a comparison, the mortality rate of the 1918 influenza virus, which killed millions and millions of individuals, was about 2.5%. There was a debate in the influenza virology community regarding whether the H5N1 virus could become transmissible. Eventually, 2 laboratories, the Fouchier Lab in the Netherlands and the Kawaoka Lab in the United States and Japan, decided to test this experimentally. Using slightly different approaches, The two labs first engineered the virus to allow it to bind to cellular receptors that are present in human airways. These receptors differ from the receptors found in avian species. They then took those viruses and passaged them through ferrets, which are a widely used experimental animal model for influenza pathogenicity. Both labs were able to isolate viruses that could be spread through the air from ferret to ferret. In other words, these viruses had a gain of function, allowing them to be transmitted from one mammal to another. When the labs wrote up the results and submitted them for publication in the fall of 2011, the US government became aware of the work, and because the research was funded by the United States National Institutes of Health, or NIH, the government asked the NSABB to make recommendations about the wisdom of publishing the data. After extensive discussions, including with the authors, the NSABB recommended against full publication because of the concern that someone wishing to do harm could reconstruct the viruses and use them maliciously, and the thinking that this outweighed the potential benefits. Rather, it recommended that the fact that the virus could become transmissible should be published without disclosing the nature of the mutations involved. There was an outcry from the researchers. The journals agreed to the NSABB recommendation only if the United States government would ensure that those details be made available to those in the public health community who needed to be aware of the information, as well as scientists who might wish to build on that information. The World Health Organization convened a meeting of influenza experts who concluded that the papers should be published in their entirety. Meantime, the influenza research community imposed a moratorium on itself to allow for the issue of whether these types of experiments should continue and how so to be debated. One of the senior authors, Ron Fouchier, claimed that he had been misunderstood when he publicly stated that these lab-derived viruses were very lethal. In the lay press, the New York Times called the virus a doomsday machine. Eventually, a decision was made to allow the authors to submit revised manuscripts that more clearly described the safety precautions that were taken, and also in which the Fouchier group gave a more accurate depiction of the true virulence of the virus they had isolated. These manuscripts were once again reviewed by the NSABB, which was also told by the leadership of the US National Institutes of Health that selective sharing of the results was not feasible under US law. The board also heard from the authors, the editors of the journals, and others, including members of the public health community from around the world. At the end of this second round of discussions, the NSABB voted to recommend publication of both revised manuscripts. The vote for the Kawaoka paper was unanimous, but that for the Fouchier paper was split. Both papers were published shortly thereafter. There were a number of things that occurred after these decisions were made. First, the US government implemented a policy regarding the review of research with dual-use research of concern potential at an earlier stage in the process, beginning with the submission of grant proposals to the NIH. The US government has also proposed a policy for review of such research by the institutions at which the work is being performed. At the time that this video was recorded, that institutional policy had not yet been formalized, however. Second, while the charge of NSABB was to advise on biosecurity concerns that might be raised by these manuscripts, there are also clear biosafety issues that are raised by the H5N1 experiments. While the authors assured the world that they used appropriate biocontainment, a number of people have pointed out that if similar research is carried out at an increasing number of sites globally, it may only be a matter of time before there is a laboratory accident that results in release of a dangerous pathogen. This assumption is based on the fact that accidents at biosafety level 3 laboratories are more common than one would like to occur. Additional manuscripts dealing with H5N1 influenza and other pathogens, which clearly fall within the realm of dual-use research of concern, have been published since then without nearly the same level of scrutiny. In January of 2013, the influenza research community decided to end its self-imposed moratorium. Now, some have argued that a stated purpose of the moratorium, to foster a broad discussion of how to proceed with similar experiments on influenza, was not achieved. Along these lines, some scientists and others have postulated that gain-of-function experiments with the potential to create highly pathogenic organisms might even be unethical, and have called for discussions along those lines. Despite these calls for caution, however, gain-of-function experiments on influenza are moving forward. So what do we have to learn from the H5N1 controversy? The first critical lesson is that the publication stage is too late in the process for discussions to begin. While the authors did get approval from their respective institutions' biosafety review committees, given the high stakes of this type of research, one can't help but wonder whether a broader discussion early on would have been appropriate. I certainly think that is the case. For example, some very knowledgeable individuals have argued that the proposed benefits of the research were overstated, and there also are the ethical concerns that I just mentioned. A second important lesson is that the debate was tarnished to some degree by opinions, in some instances voiced by leaders in the field that were not necessarily supported by data. In addition, some have argued that the NSABB was not given all the information it needed to make a well-informed decision. In the future, then, I think it is incumbent on all involved to have all the facts, and only the facts, available as these discussions are taking place. Finally, despite ongoing worldwide efforts to raise awareness of the dual-use issue, it was clear from the way that the H5N1 controversy played out that a majority of the scientific community was still in the dark. Let's now come back to the more general discussion of responsible science. If we are to foster a culture of responsibility, what are some of the factors that need to be considered? First, we have to think about why we do research. I think there are two general goals to research in the life sciences. One is the desire to understand the living world around us, why biological systems behave the way that they do. Of course, this is called basic knowledge. The second goal is the desire to make our world a better place by understanding health and disease, and by doing what we can to either prevent diseases in the first place or deal with them efficiently once they occur. I would emphasize that this is true whether we are concerned about human disease or diseases of animals or of plants. The second thing we need to consider is something that transcends science, namely our rights as individuals in most parts of the world to express ourselves freely. Some have argued that one can do any experiments one wants to because this is a form of free speech. A specific type of free speech that has been invoked by people working at universities is that of academic freedom. It is my opinion that these quote-unquote free speech rights do not apply when the results of free speech or academic freedom, in this case being the experimental data, may cause harm. Now, this is not unheard of. Here in the United States, for example, our Supreme Court has ruled that so-called hate speech, or speech that is terribly offensive against certain persons, is not protected by our constitutional right to speak freely. Of course, what makes communication potentially especially dangerous in the 21st century is the ease with which people can speak, namely through the internet. Information that once took weeks or months to be disseminated is now available almost instantaneously. This rapidity of communication goes hand in hand with the speed at which science progresses. Technology is always advancing, and the financial barriers to performing life sciences research are very low compared to other technologies with which someone might attempt to cause harm. I would like to conclude this video with a few slides that emphasize some final thoughts about the research enterprise and why we as scientists must act responsibly. There are many groups that have stakes in the research enterprise. The scientists themselves, governments in whose countries the research is performed, policymakers who determine how public dollars should be invested in research, commercial entities that may seek to profit from research results, and security communities whose job it is to worry about possible malfeasance, just to name a few. But I would argue that the most important stakeholder is the public as a whole. There are two very simple reasons for that. First, our research benefits the worldwide public. As I just mentioned, our work is driven towards making them healthier, as well as the food that they eat and the environment in which they live. Second, in many, if not most countries, it is the public that pays the bills for the research that gets performed through taxation or through spending of dollars that could go towards other items. Therefore, it is our duty to show the public that we are acting responsibly as we carry out our incredibly important mission. Unfortunately, and perhaps especially so in the United States, science is not always highly respected. While polls overwhelmingly demonstrate that the public thinks scientific research is important, their perception of scientists is often not as positive. Scientists are often portrayed in the movies or TV or books as being crazy. Think about some of the villains in James Bond movies, for example. We as scientists don't always help our cause. We can often come across as aloof or disinterested in what the non-scientist thinks. We sometimes have the attitude that we know what's best for science and don't have to listen to what others have to say. I believe that this is a damaging way to behave. Finally, science is sometimes viewed as being wrong because it appears to contradict religious beliefs. Some of the current arguments against whether climate change is real fall into that category. I think that the public forgets how dependent it is on science and technology. Yet, somehow the results of scientific experiments are sometimes not accepted. There seems to be a gap in logic here. We as scientists must be aware of the potential outcomes of bad behavior, especially if that behavior leads to lapses in biosafety or biosecurity. First, there would be justified mistrust of the scientific community that would go well beyond the current doubts about science that I just discussed. This would make the job of those who are working hard to build public confidence and support for scientific research much more difficult. Second, governments might react by imposing tough restrictions on research or by limiting funding. Such regulation could seriously hamper legitimate science. Even science that falls outside the realm of potential misuse could end up being regulated. Thus, there could be harm to the broader life science enterprise. In addition, unilateral regulation would just change where certain types of science get done. For example, when the Bush administration in the US put tight restrictions on stem cell research, a lot of that research moved outside the United States. And of course, we must consider the harm that could be done to the public, both directly in the short term as well as indirectly in the long term. A short-term effect might be due to the accidental release of a dangerous pathogen from a laboratory, resulting in disease and possibly death. The long-term effects are related to the first 2 points I made, in that slowing research down would undoubtedly slow the discovery of much-needed vaccines, therapeutics, and diagnostics. So what can scientists do to improve public perceptions and demonstrate that we understand our responsibility to the public. First, we need to hold ourselves to the highest ethical standards. While this video is focused on potential misuse of research results, of course we must also pay attention to misconduct issues such as plagiarism, data manipulation, and the like. There have been calls for codes of conduct for life scientists, and a number of groups have put together proposals regarding how to approach the development of such codes. Codes are not unheard of in science and technology. Engineers subscribe to codes, and of course physicians take the Hippocratic Oath. The bottom line is that we can talk all we want about how responsible we are, but at the end of the day we must back up that talk with action. Second, we need to be sure that the scientific process is open and transparent. Secrecy will only breed mistrust, even if that secrecy is only perceived by the public. And now by secrecy, I'm not talking about classified research being carried out in government laboratories somewhere. Rather, I'm talking about the tendency of scientists to have the attitude that I mentioned earlier, in which we come across to the public as if we are saying, "Trust us, we scientists know what's best for science." Let's take the time and make an effort to openly discuss what we are doing and why, in terms that scientists and non-scientists alike can understand. I think we have another tendency, at least in the United States, to bemoan how uninformed the average person is about science. I think it's our job to make what we do understandable. And this leads to my third point, which is that we have to engage in an ongoing dialogue with the public. In the end, scientists cannot bury their heads in the sand. It is sometimes easy to say, "That's not my problem," or "I don't have to worry about someone misusing my results because it's just not going to happen." We cannot assume that our being responsible is implicit in the way that we are viewed by the public. I believe strongly that we must actively demonstrate that we are doing everything we can to minimize the possibility that our actions as scientists might lead to harm. I think that in addition to interacting with the public, as we just discussed, we also need to raise awareness among our colleagues that in addition to performing our research day in and day out, we have an equally crucial role in communicating what we do and, just as importantly, how and why we do it to the public at large. I would urge all of you to get your colleagues in your departments and institutions on board with the idea that this is an important goal. I also think that we must make our case every chance we get. In the United States, there is an advocacy group called Research America, whose goal is to raise awareness of the importance of research and lobby for more investment in research. Their president, Mary Woolley, has some great advice. She says that if a scientist meets someone and that person asks, "Who do you work for?" your response ought to be, "I work for you." Then you can use that as the start of a conversation about the importance of scientific research to the public. The message must be that the scientific community, through our research, is dedicated solely to the betterment of mankind. I hope that you have found this discussion of how we might foster a culture of responsibility to be helpful. Thank you very much for your attention.